ask Fernando to please share his screen and start.
Okay, so let me share my screen. So guys, welcome. I hope you are safe and your families. This is, new times. And as Tura said, I am here to tell you a little bit, to introduce this topic that, we have been addressing in JPEG. And as you know, it is a named JPEG-based media storage. Okay, so after me, you will hear the real experts. You know, I've been in this area not that long, and as you can see, this presentation has been prepared together with Turaj because this ad hoc group in JPEG was launched by the two of us, although the idea was coming from Turaj, and I'm basically helping him to shape this new activity. Okay, so the context is very obvious. The world is full of data, is full of bits, and many of these data, of course, is multimedia data. We have a lot of images, and the images are very dear to JPEG. And of course, we have a lot of video, a lot of audio, a lot of speech, a lot of everything. Okay, so we have tons and tons of data, tons and tons of bits, usually. And of course, this data has to be stored. And of course, the world is looking for efficient ways to store all this multimedia data, sometimes with the requirement that the storage is very long. So we have some world heritage in terms of data that should last for very long. And so this type of requirement may be important for the workshop of today. Of course, you know that JPEG is very familiar to multimedia and very familiar to storage, and we have a family of standards that, of course, involves a lot of image codecs, as you can imagine, from the old good JPEG to the very recent JPEG XL standard. Of course, we have also some system standards. In order we can use these codecs in an efficient way in multiple application domains. And as we know, we have also a standard on quality evaluation that it is this AIC. Okay, so it's this JPEG universe that somehow has been addressing needs in the storage domain, and we want to continue to do that. And that's why we have started this activity related to DNA storage. Of course, we have a long story, as I said, in storage. JPEG, I mean, the old JPEG standard has been used, for a lot of images around the world. And these images are, of course, stored in many places, with different qualities, with different purposes, with different durations and temporal durations in mind. Of course, this chart is old, but it's just showing you that there is an exponential trend on the usage of the old JPEG standard that keeps really exploring in terms of usage around the world. In the storage domain, it's also important to mention JPEG 2000. As you know, JPEG 2000 has some nice features, notably related to scalability. And it's an image codec that it's really much used, especially in archives, for example, national archives, and also in many applications like you see here, medicine, digital cinema. So you have really a lot of content that should be stored, and probably some of it should be stored for a long time, coded with JPEG 2000. But of course, in JPEG, the past is the past. We love that JPEG standards are still used after 30 years, like our old JPEG standard, but we have to look to the future. And as you can imagine, there are new things coming every day and new technologies, and especially new technologies for storage and storage media. Of course, it's very important for JPEG. And so the context of this workshop, it's really exploration and trying to understand what is really new in terms of storage. And in this case, in DNA-based storage, that is a kind of, when I heard about it the first time, it's a kind of surprising, what is this DNA-based storage? Okay, let me tell you some basics. Okay, so we all know DNA. DNA is the molecule of life. Living organisms, they have DNA that basically carries the genetic information of these organisms. So the genetic instructions, even for the evolution and for reproduction, they are basically carried by the DNA information. But as you know, DNA is made basically, it is a kind of alignment of simple units that when they are put in a certain order, they basically give this genetic information, okay? So basically the sequence of the units that you use in DNA carries information, well, very much in the same way that there is information carried by the letters of the words, the sequence of letters and words in a text. Okay, so the DNA is basically a sequence of basic units that in their order carry some information. So you could think that we could create artificial DNA molecules. So now we are not speaking about the DNA from living organisms, but we could think about creating artificially DNA molecules where we control the order of these units. And by controlling the order of this unit, we basically store some sequence of information, some media message, okay? So the idea is that DNA molecules, in this case, artificially created DNA molecules, could be used to store information, and in this case, information related to images and video and audio. Okay, so what are the basic units in the DNA building of a molecule? So there are basically four DNA bases, or nucleotides, as you prefer, and they are called adenine, guanine, the larger ones, and cytosine and thymine, the smaller ones. Well, the small is the pyrimidine, but we have basically four units, which are adenine, guanine, cytosine, and thymine. So they are typically labeled as A, G, C, and T. So the basic idea here is that we could store media information in an artificial DNA molecule by controlling the sequence of these basic units. Okay, so that's the basic idea. So making it very simple, what does it mean? Well, it means that we would not be using our usual binary system where we basically transform our images into a sequence of bits, but we would be using another type of system, in this case of quaternary system, with four bases, A, G, C, and T, that are the basic units in a DNA molecule, and we would create streams that are like this, A, G, T, C, T, T, okay, blah, 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 okay? So that's the basic idea. So we would be basically storing this information in a molecule by controlling the sequence of the basic units, and the basic units are now bits 0 and 1. They are elements, units in this quaternary system, A, G, C, and T. So that's the basic idea. Why is this so interesting? Well, there are at least three characteristics that make this very interesting. Well, first of all, it seems that the density of the storage, so the amount of information that you can store in a cubic centimeter, this type of measure, it's very, very high, okay? And of course, if you have a lot of data, the fact that this storage is very dense, it's very interesting because, of course, we want to minimize the size of the storage. It seems that it's also very stable. And you see here an example where we could recover DNA information from a horse that has been living 500,000 years ago. Okay, so it's very stable. And this is important for many applications, for the heritage, for the media heritage of mankind. We need a storage that can, be very long in time. And finally, it seems it's also very energy friendly, ecologically friendly, because you don't need a lot of energy. And these features, high density, very stable, and not much energy, they are really a trio that it's extremely interesting, especially comparing also with existing technologies. Some of them are not probably dense enough, they are not stable enough, and some of them require a lot of energy. So the basic features of this type of technology would be very, very interesting. So when you think about this type of technology, we have basically a process that has these steps that we can see here. So the first step, of course, would be an encoding step. So you will have to represent the data in a sequence of these units. So not in a sequence of zeros and ones, but a sequence of these basic units A, G, T, and C. Okay, so this is the encoding part, and it is very similar to what we did in the past for a binary system. So it would have to be the same type of thing for quaternary system. Of course, when you have this sequence, so when you have the sequence, you will have to synthesize the DNA molecule. Okay, so it's a process where you create, it's a biochemical process where you create an artificial DNA molecule with the sequence of units that serves to code the images, for example, that you want. And finally, these molecules, they have to be encapsulated in some physical container. It can be a tube that we put in a refrigerator, or today probably, or it can be something more sophisticated in the future. But in these three first steps, you encode, you create the artificial DNA molecule, and you put it in some container where it can stay, in theory, for a very, very long time. Of course, after you have to do the opposite process. And the opposite process is the so-called release. So you have to, when you want to decode, you have to go to this container and basically extract the molecules from this container. And I put here this thermal damage simulation because in this process of encapsulation, putting the molecules in this container, or in the releasing phase and the sequencing after, you can have errors, you can have noise, okay? So in the release phase, you take out the molecules from this container, and in the sequencing phase, you read out the sequence of A, G, T, and C units from these molecules. And finally, when you have in your hand the sequence of A, G, T, and C, you basically decode your data. Okay, so it's basically a process where there is a coding part, you create the molecules, you put them in some container, and after you do the opposite. Okay, so this is a graphical representation of this architecture, of this process. Okay, so in this process, there is one question that may be very fundamental, which is, okay, how do we go to these 4 units domain? How you go to the CGTA? Okay, so the first idea that you can have is to say, okay, I have my content today, for example, a JPEG image, where it is represented in zeros and ones, and I can transcode the zeros and ones into C, G, T, and A. Okay, so it's basically a transcoding approach. We start from the content that we have today with a binary representation, and we have a kind of transcoding model from binary units to quaternary units, okay? Of course, when you do this, in a way, it is friendly to the content that already exists, but in fact, when in the new quaternary representation, you may not be as efficient as if you do it directly. So in this type of architecture, we have what we call the coding architecture, because we don't go to the binary representation. Okay, so we don't do the entropy coding part in the binary representation, but you can go directly from the symbols, and the symbols can be quantized DCT coefficients, or it can be quantized wavelet coefficients, or can be anything else. You go from your symbols, and instead of doing the binary entropy coding, and after the transcoding, you go directly from the symbols with the probabilities to the quaternary representation in C, G, T, and A. Okay, so in theory, this can be more efficient because you don't have this intermediate binary step. You go directly to the quaternary representation, although you may lose a little bit of compatibility with the existing binary world. Okay, so one of the things that we have been discussing in the ad hoc group is precisely what type of architecture is relevant. And it seems that the preliminary conclusion is that maybe both are interesting. Maybe both are relevant for different application domains. What are the challenges of this thing? So it's all beautiful. Well, of course, this is very new. It's emerging. And the challenges are a few challenges. I will highlight 4 challenges. So the first one is the cost. Today is quite expensive to put these quaternary elements, our information, and create the DNA molecules, and to create them with the sequence of units that we want, and also the reading of this information from the molecules. This is expensive. Okay, of course, we expect that with time it will become less expensive, but today, the cost is an issue. like the speed. This is not a process that can be very fast. So today, the writing and the reading speed, it's an issue. Another issue is the errors. This biochemical process may have some thermal degradation or other types of degradation. So there are errors in the process, especially if you use certain combinations of units. For example, it's not a good idea to have many units of the same type, many A, T, 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 okay? These type of combinations are not good from the error point of view. So we should avoid them in our coding process. And of course, this has to be controlled. And finally, the random access. If you basically store some information in DNA, it would be nice that you don't have to decode the full information to access a part of it. So the random access is a very important requirement in multimedia. And so it would mean that in DNA storage, it should be possible to read part of the information and not the full thing to access a part of it. So this is the type of challenges that we have. I'm not saying that these are the only challenges, but these challenges we are already important enough to capture our attention. And what are the type of use cases? Of course, as I said, it's very much about storage, okay? But in our discussions, we already identified at least 2 cases that are interesting, and they are a little bit different. Well, first of all, this long-term media preservation case, okay? So like the National Archives, where you put very basic information for a country, for a nation, for mankind, and you want to preserve it for a very long time, okay? So longevity is an issue, okay? And probably because we want to preserve the information, quality degradation is also an issue. We don't want much quality degradation. And maybe even lossless coding is an issue. Okay, so because it's such a basic storage. But on the opposite sides, we may have, for example, the cold media storage of social networks. You know that in social networks, some of information, especially from last year, 2 years ago, it's not so much accessed. Well, it should be available, but it can be accessed in the so-called cold storage, because the access can be a little bit less quick or less easy. Okay. In this case, maybe, if it's a Facebook image, if it's an Instagram, maybe you can afford some quality degradation. But these 2 cases, they represent basically usage that seem to be very relevant for this type of technology. So a lot of data, we need high density, duration in some cases, okay? And of course, if you can be ecologically friendly, it's of course very, 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 very important. So what type of requirements may we need for this type of system and this type of coding? If you go on the coding architecture, we will have to code our content, our images, for example, directly into these ACGT units and not using our usual binary representation. So of course, you see the requirements, and some of them are very expectable. Of course, we want compression efficiency. We want to put as much data in these molecules. Of course, random access, I told you it's important. Random access is very important for multimedia. Error resilience. I told you there are some errors in this process, so we may need to be error resilient to the errors in this process. Scalability may be a relevant requirement. Okay, so we want to decode an image, but as for JPEG 2000, I want images maybe with different resolutions in layers. Okay, and we have some new requirements that are interesting. Ambiguity. So in this process where we put the information in the molecules and we have to read out of the molecules, we should not have any ambiguity in this process. We cannot read anything that can be a 1 and a 0. It has to be univocous, unambiguous. And the biological constraints, I told you, we need a standard that takes into account the biological and biochemical constraints of this process. For example, not too many units, not too many nucleotides of the same type in sequence. Okay, so this will be very important for the codec. And finally, you may even want a requirement in terms of artificial recognition. I read some DNA sequence and I know it's artificial, it's not from a living body. Maybe we want to create sequences that can immediately recognize as artificial and not from a living organism. So this is just examples, okay? This is a very early process, but as you can see, some of the examples are the usual ones, some of the requirements are the usual ones, and some of the requirements are new ones. What are the next steps? Well, we are, with this workshop, trying to advertise this activity, okay? We need expertise. We need critical mass. To have a good standard, we need people, we need experts, we need brains, okay? Of course, we are organizing workshops like this one because we want to discuss with the experts, like the two speakers after me. We have to understand the market needs. We have to understand, okay, what is the stage of technology, okay? How mature it is or it is not. We have to identify use cases and requirements, like the first ones that I showed you. Potential technologies, what are the technological solutions? What type of solutions are already available in the literature? Is there enough technology to think about making a standard out of it? What is the time to the market? Is it something very early, or it's already something that can be mature maybe in 5 years? I don't know. We will discuss that. And of course, you know, we can do some very basic things, and probably the most basic one could be following the transcoding architecture. When you go from our binary representations of today, and we have some efficient way of transforming the binary representation into the quaternary representation. And later on, we can follow the coding architecture where we go directly from the symbolic representation to the quaternary representation. So there are a lot of things, exciting things to be discussed. So my presentation is basically a teaser for you. So if you find this interesting, this is starting. So join us, and together we will create some great JPEG standard for this application domain and for this technology. So if you have any questions, or if the chair wants me to say something, of course I will be pleased to answer your comments and your questions. Touraj, the floor is yours.
Thank you very much, Fernando. I propose, because of the interest of time, we move to the next presentation. We leave the questions for the wrap-up session. Now, okay. Nothing will prevent you, please, anybody who has a question, to actually enter your question already on the chat window. This way we can organize them, and also the speakers can think about it. So let's, without any further transition, just move to Mark. Mark, the floor is yours, and I guess you just need to share your screen.
Is my screen shared? I don't see.
Not yet. I see you.
You, but not your screen. No, something is coming. Yes, now we see.
Something is coming. Is it a good presentation? Yes, I guess it is. Okay. So, okay. So, thank you, Touraj, and thank you also, Fernando, to invite me to give this presentation today. It's a pleasure for me to participate also to this JPEG meeting and to this group, this new group on DNA coding. So is it okay? Do you hear me? It's okay?
Yes, we hear you perfect.
Yeah? Okay, great, thank you. So the title of my presentation is Image Coding for Long-Term Storage on Synthetic DNA. So, oops. So this is, of course, a joint work, first with my two PhD students, so Mel Promeni and Eva, who are here today, attending the meeting also. And a joint work with Raja, who will give a presentation after. So Raja is assistant professor from URICOM. And also we are working with some biologists, and especially Pascal Barbry, who is a research director at the IPMC, which is a biology lab for biology in Sophia Antipolis. So I want to start this presentation by just this observation. And maybe I guess you are aware about that, but it is interesting to recall is that in 2025, the amount of data, of digital data that we will have on Earth is estimated to be 175 zettabyte. So this means that we will have 5 times more data than today. And if one were able to store this data on Blu-ray disc, for example, you will get 23 stacks of disk that has that have the distance of that is okay. You can have 24 stack of disk with the height of the distance from the Earth to the Moon. So this is something very huge. And we have to find some solution to store this data. So a significant fraction of this data is called cold. So this means that this data is infrequently accessed. So for example, old photographs stored by Facebook are one example. And the growth of this data is a 60% rate. And however, the improvement of the storage density of the hard disk drive is 20% per year. So this means that the amount of data that we are producing is much higher than the capacity of the hard disk drive we have, or the storage solution we have for storing this data. And also the storage media that we are using, for example, hard disk drive and tape, have a limited lifetime. So we need to replicate and to copy data on new hard disk drive or tapes every 5 or 20 years to preserve this information. So this has a cost, and in terms of money, in terms of energy, and this is not so. This is bad. Of course, there are emerging new solutions, new storage solutions, and people are interested, for example, in using silica glass for storing data. So the interest of silica glass, for example, is that this is very hard to destroy. So you can store data on this kind of device, and okay, this is very strong. You can baking in oven at 500 degrees, and there is no destruction of this glass. However, the capacity of storage is not compact. For example, on this example, the Superman movie of 1978 was stored on this silica glass, which has a size of 7.5 by 7.5 centimeters. Okay, so this is not very compact, but very robust. So it's you can store for a long time. Also, the access of the data is fast in the sense that we are able to read the data back directly on the silica glass more or less easily. Okay, so oops. If we consider DNA, it is estimated theoretically that we can store on a single gram of DNA 215 petabytes of data. So meaning that DNA is ultra compact, and also as Fernando told us previously, DNA can last hundreds of thousands of years if it kept in cool and dry place. The fact is that today, for when we want to synthesize DNA, this has a cost, and roughly, if you want to synthesize 200 nucleotides, this has a cost of $1 more or less to synthesize 100 nucleotides. So this means that if we want to encode data, we cannot encode data as they are. We need to do some compression for reducing the amount of data we have to minimize the cost of synthesis. So how to store digital data in synthetic DNA? So the main workflow is the following. So if you consider data at the input, the idea is to encode the data in using a quaternary alphabet, ATCG, as presented previously by Fernando. And so once this is done, synthesis can be done. And once we have the synthetic nucleotides, we can store them in such in a specific device. And this device, which are a capsule, I have an example of the capsule later, so I will show you the picture. So they are stored in a specific capsule that preserves the DNA from water and oxygen. And okay, this can be stored for a long time. So the day we want to recover the data F, we must do the sequencing of this DNA. So, and we can recover the sequence of ATCGs that has been generated here, and thanks to a decoder, recover the original data that we have stored. The fact is that sequencing introduces errors. And mainly, there are two kinds of errors: substitutions of a nucleotide, which means that we replace A by C, for example, or T by G, or G by C. So we have a replacement of nucleotide, and also we can have indels. We call indels insertion or deletion of nucleotides, meaning that we can lose one nucleotide, or we can insert one nucleotide or more. And we can have more insertions than deletion, or more deletions than insertion, or the equal amount of insertion of deletion. So if you don't have the equal amount of insertion and deletions, or if you don't have the equal amount, you have a sequence of the sequence you are decoding don't have the same size as the sequence you have encoded. On the contrary, if you have the same amounts of insertion and deletions, it's like substitution. So you have the same amount of nucleotides after sequencing. So this is something which is a little bit complicated to deal with. Okay, so the basic idea, as was presented by Fernando, the basic idea of doing DNA coding is to transcode. So it's to convert a sequence of bits in sequence of nucleotides. So to go from a binary stream to a quaternary one. And so most of people in the state of the art are doing that, and we will discuss this a little bit later. The fact is that it's not so easy to do that, because since synthesis and sequencing are introduced noise, and especially sequencing, we must add some biological constraint on the code we are constructing. And for example, we cannot generate homopolymer runs. So an homopolymer is a consecutive occurrence longer than 3 nucleotides, such as A, for example, or T. So this is not in general good for the sequencer. Also, the content, the amount of GC of G and C nucleotide in the stream must be lower than the amount of A and T. Also, we should avoid pattern repetitions. For example, if you have a code ATC, we cannot. It's preferable to avoid ATC in the stream. Another constraint, which is linked to the synthesis, is that today, if we want to do synthesis without a lot of errors, or without error, the oligo should not be longer than more or less 300 nucleotides. So this means that the sequence, the DNA sequence we are generating when encoding data must be cut into small pieces, into chunks, and to be synthesized. And then after the sequencing, those chunks must be reassembled to reconstitute the data. Okay, state of the art. So, okay, the idea of the storage on DNA is not very new, but the pioneer works are recent, and especially the works of the first one doing a synthesis, synthesis and the sequencing is Church in the 2012, and then some. I don't want to say several because there are not several people working on that today, but some people were interested to do that. And there are some interesting papers since 2012 until 2017. And roughly, Church, the first who made the first step in DNA storage, introduced a very simple encoding, which has a low compression potential. And also, this solution can only be applied to binary input data. This means that you can only do transcoding. But the idea is that this work provided important information on sequencing error. So this is a pioneer work, which is very interesting because it helps for when we want to study the sequencing error. Then in 2013, Goldman introduced another procedure which is based on the ternary Huffman. And each trait of the ternary Huffman is encoded to one nucleotide, avoiding the previous nucleotides that had been used. So this algorithm is interesting because it improves the quality of the sequencing, because it avoids homopolymers, and also is efficient in terms of coding, of compression, but it introduces pattern repetition, especially when you are encoding quantized signal or spark signals, for example, when you are encoding wavelet coefficients, for example. Bornhold improves the works of Goldman, so it improves the performance of coding. Okay, by avoiding some process that Goldman introduced. So I will not enter in the detail here. In 2015, Grass proposed to use forward error correction with Reed-Solomon codes. So this solution has interesting coding potential, allow, of course, error protection, avoids homopolymer, but can only be applied to binary input data, so can only be used for transcoding. Blawat in 2016 introduced a solution which is efficient, allow correction of some types of sequencing error, avoid patterns, but cannot be applied to can only be applied to binary input data. And also, it develops a solution for packets of 8 bits that are encoded in 5 nucleotides, and it cannot be extended to the encoding of more than on packets with size more than 8 bits. And finally, Ehrlich in 2017 used Fontaine code for robust encoding. So it's a very high efficiency algorithm encoder. Of course, only applicable to binary information also, and very expensive, but very expensive in computational cost. We recently proposed in my group also a coding solution. We call the solution PairCode, which is a low complexity solution to construct biologically constrained quaternary code that I will detail now. Okay, so PairCode is a code that allows to avoid homopolymer and also allows to control the amount of GC compared to the amount of AT in the sequence. So it is based on the construction of a code, based on 2 dictionaries, C1 and C2. And the first dictionary, C1, contain doublets of nucleotide. Those doublets are chosen such that when assembling together, we are not able to generate homopolymers. So when you want to construct a code with an even length, you can join together codes from the code words from the code C1. Now, if you want to generate odd length code words, you can add at the end of the code that you are generating that you have generated with the C1 code. You can add at the end of the code words a symbol from C2, so ATCRG. So using this algorithm, you are able to generate even length and odd length codes. The fact is that you are not able to generate all the possible codes that the ATCG alphabet can provide because you introduce a constraint which is avoiding homopolymers. So the assets and weakness of this code. So this is a fixed length code. So this means that when we are encoding, we haven't a variable length code. So why we did that? We did that to be able to decode code word even when there is noise in the sequence. So it's a constraint that we introduce, and it was our objective. So of course, using fixed length code, we will be less efficient in terms of coding potential, but we will be more robust to sequencing error, allowing easier error correction, of course. This code can be applied to any type of input data because it's not limited to, our code is not limited to binary sequence. So we can encode binary or not binary, which is different from most of the solution of the state of the art I presented before. So it is simple in terms of computational cost, but it contains extra redundancy. And we use this extra redundancy to avoid pattern repetition. So what is extra redundancy? So it's more or less explained here. So sigma is the code book containing the code words that we use for the quantization, so the symbols. And C star contains the code that the DNA code that we are using. So it's a fixed length code. So what happened is, let's suppose that K is the size of my dictionary sigma, and capital L is the size of my code C star. It is obvious that according to the code we are constructing, we cannot exactly fit the value of K. So we generate, in general, more codes than necessary for encoding K. So generally, capital L is bigger than K. So we have some code words that are remaining here and which are not in general used. So what happened is that instead of wasting those code words, we are using them and reassigning them to the most probable code words of the quantization dictionary sigma, allowing to have for the most frequent code words in the sigma, allowing to have a double representation, a double code, such that if this value appears several times, like for example in the Weblet subbound after quantization, if the quantization value appears several times, we switch between the two codes, the two representation, for encoding this quantization value. So this is a way for avoiding pattern repetition and also to avoid wasting this remaining code at the end of that are generated at the end of the codebook C star. We can adjust also the value of L to have different solution for assigning this double representation, so the extra code for the code words in sigma. We have compared this solution to the algorithm of Goldman. So we have transcoded the JPEG and JPEG 2000 bitstream with pair code and with Goldman on the LEDA image on size 512 and 512. So in black here, we can see the results of the Goldman encoder for the JPEG stream on the left and the JPEG 2000 stream on the right. So this is pQSNR versus rate. So the rate here is expressed in nucleotide per pixel. We can express also the rate in bits per nucleotide, which is more a compression power than a rate. And we compare this to our encoder. So in blue, so per code applied to the as a transcoder without avoiding patterns. So here we have patterns, and in red we avoid patterns. So introducing the constraint of patterns, of course, decrease the performance of the encoder, but still we are close of the performance of Goldman, which doesn't take care about the patterns. And we have the same results for JPEG 2000. So what is interesting here is that with a simple fixed length code, we have more or less the same performance of a code, the Goldman code, which includes Huffman, a ternary Huffman for doing the encoding. So we generate, which take into consideration the statistic of the source. If we compare our solution here on the last columns of the table with the state of the art, so the other solution. So here on the first line, we have the size of the data that have been encoded. So those results are taken from papers. So people were encoding different data. So the data is not the same here. But anyway, what is encoded, what is transcoded is bit rate. So really, they don't care about what kind of data they are encoding. So we did the same here. So the amount of data that we're encoding and the coding potential here in terms of bits per nucleotide. So Church is, okay, so our solution provides 1.6 bits per nucleotide. Church is providing 1, so it's 1-to-1 conversion. And we can see that the other people are around 1.6 or until 1.98, so 2 bits per nucleotide. We can see that here some of the methods have error correction, others no. In our case, we don't have error correction. We have some kind of error correction, so as a post-processing, but the code is not designed to be error-free. Okay, so some of the methods are able to have a full recovery of the data, others no, because of the noise introduced due to the sequencing. So, however, if we are doing transcoding like that, we don't take into consideration the nature of the input data. So here, for example, we have JPEG. The process that have been done is the following: we have an image, JPEG coding, so we have binary, and then we transcode with different method. And so here, the transcoder don't care about the data, the nature of the data as input. It's just transcoding. So we propose, and like previous work, to define an algorithm, an encoding algorithm, which deals for image storage into DNA, so specific to image, because state of the art is not specific to image. They are taken a binary stream that can come from text, audio, or whatever. So the algorithm we developed is specific for images. So we are performing a direct quaternary encoding without converting first in binary. We are directly coding in the quaternary, and the objective is to take into account the nature of the considered data. Okay, and the idea here, by introducing the quaternary code encoding inside the system, is to be able to control the compression rate. So to optimize the trade-off between the quality of the decoded image and the cost. Because if you remember, when you are doing transcoding, you are not controlling the rate of the DNA, the size of the DNA sequence you have after transcoding. So you cannot control the cost of the DNA, I mean the synthesis cost. So if you want to control the cost of the synthesis and also the quality of the image, it is interesting to insert the coding, the DNA coding inside the compression system. And so the proposed workflow is the following. So in blue is the coders and the decoders. So, and in red is the biological part of the system, of the workflow. So if you have an image F, we propose to use a wavelet transform and to quantize the coefficients of the wavelet transform. Thanks to, we define, we optimize the quantizer thanks to a nucleotide allocation across the subbound. And then after quantization, we encode thanks to our pair code, our DNA code, we encode to DNA. And we have also formatting. So if we forget what happened at the middle, then if we have the DNA code here, I am able to do the DNA decoding and reconstruction. And thanks to the wavelet, the inverse wavelet transform, I can recover an approximation of F. And I have minimized the distortion between F and F hat, thanks to the nucleotide allocation. Okay, now in the middle, we have DNA synthesis, which is a biological process. So after creating the DNA code, the DNA can be stored. And then one day, if we want to decode, you need to do the sequencing. And so before sequencing, there are some operations called the PCR amplification, polymerase chain reaction, PCR, that allows to add redundancy and to make the DNA, the synthetic DNA, more robust to sequencing noise. After that, we have to select the oligo that has less errors, let's say. Here we can have PCR amplification can amplify to millions of oligos. So I forgot to say that when you are synthesizing the DNA, you are synthesizing oligos. So it's, for example, 300 nucleotides. If you have a stream of 1 million nucleotides, you cannot synthesize 1 million once. You have to cut in chunks of 300, for example, and then synthesize each 300 nucleotides. And those 300 nucleotides, we call them oligo. So as the output, okay, when before doing the sequencing, we amplify those oligos. So we have millions of oligos doing the sequencing, and then we have to select the best oligos for doing the DNA decoding and the reconstruction of the signal. Because, as I said, sequencing is introducing errors. So our solution that we implemented, we know that even if we are optimizing the rate distortion, trade-off rate distortion, we are not very efficient in terms of compression. Why? Because we are not using variable length coding. We are just using scalar quantization or vector quantization. Also, we did work on vector quantization and fixed length encoding. So this is not very efficient in terms of compression, but efficient in terms of robustness when we have to deal with sequencing error. So we did a wet lab experience 2 years ago to verify the feasibility of storage and perfect reconstruction using our encoding algorithm. So we synthesize, we compress 2 images of size 128 by 128 pixels into synthetic DNA. So why such small images? Because it was very expensive for doing the synthesis. For example, for synthesizing 2 images plus some data that was provided by Raja, we spent more or less 2,000 euros for synthesizing the DNA sequences. So we compress our 2 images at 2 rates, 2.68 bits per nucleotide and 1.78 bits per nucleotide with those 2 pKSNR. So the synthesis has been done and the synthetic DNA has been stored inside those capsules, which are DNA shells provided by Imagene, which is a company, French company. So we have several capsules here because we stored what we call the library, so the amplified oligos. So such kind of capsule can be stored for hundreds of years, theoretically anyway, for hundreds of years without altering, without introducing any distortion on the DNA. So how was formatted our oligo? So it's not simple simple like that, because we need we must take into consideration that the oligo that we are synthesizing have to be sequenced. So to do the sequencing, the sequencer have to know some informations, and we call this information primer. So we have primer 1 at the beginning of an oligo and primer 2 at the end of the oligo. So we need to add those informations to make the sequencer able to do sequencing. So we are losing 26 nucleotides plus 21 nucleotides to do so. And then inside, we have, of course, the payload, so the data that have been compressed, but also some information allowing to some headers and information allowing to decode the payload, and also to be able to recover the DNA strand after sequencing, because the sequencer can read in one direction or in the other direction. So we need to know, so we have some nucleotides for the sense of reading. And also we have one thing which is important, it is the offset, because the payload contains 65 nucleotides. If I encode this image of Lena, I have a lot of nucleotides, so a lot of oligo. In this case, we have 662 oligos. And we have to reassemble those oligo after sequencing to be able to reconstruct the signal. So to have an idea, the oligo is formatted like that. So we have the primer here at the beginning and here at the end. We have the payload here, and we put the header at the middle. So it's okay. This is due to the constraints that are imposed by the sequencer. And each line corresponds to an oligo containing here, I guess if I remember well, it's 130 or something, or 150 nucleotides. So we did the sequencing with this kind of machine, which is the Illumina NextSeq 500, so which introduced error, of course. And the sequencing is made more robust thanks to PCR amplification that amplifies the number of oligos. It's like cloning in multiple copy, introducing redundancy. So, okay, so after that, we were able to select oligos after sequencing. Interestingly, if we take the oligos randomly, because we have millions of oligos after sequencing, so if we take the oligos randomly, we obtain something like that. It's right-hand side. The Lena image is decoded like that. So we see that we have a lot of noise, and okay, noise is important anyway. And interestingly, if we select the most frequent copy of each oligo, and thus believe that they are more representative, the most frequent ones, we are able to recover perfectly the image that have been stored. And this is true for this image Lena and this image of the mezzanine CD. Okay, with random selection, we are not able to decode something, or something which is not good. And with selecting the more frequent, we are able to decode something which is perfect quality in here. Today, so this has been done 2 years later with our first web lab experiment. And today, 2 years later, we are the data has been sequenced again. So one capsule has been opened and sequenced again, and we want to analyze the robustness of the synthetic DNA. So this work is still in progress, and we hope that we will have good results. So proving that DNA can be stored at least for a given time, at least 2 years, without any alteration. Okay, so after the last meeting of JPEG, we were interested to see what happened with the standard JPEG codec, and we tried to insert our code in the JPEG codec. So it's a first attempt. So I don't claim that this is the best way to do things. I don't claim that this is the best result that we can have, but okay, it was just to see what can we have if we are configuring a more efficient encoder. So here in the JPEG, so this is a classical JPEG encoder, as you know, so with the DC coefficients and the AC coefficients. And so what happened is that we replace in the variable length coder here, we replace the binary Huffman by a ternary Huffman plus a Goldman encoding. So, and here it was using real frequency. So we encode the category of diff with something which depends on the probability of the category. And the value of diff has been coded using our solution, which is per code, so fixed length code. But the length of the code has been adapted to the category. The same for the AC coefficient. So here we are coding the run category duplets with a ternary Huffman and Goldman, and the value of of val is encoded using a pair code with a variable length depending on, sorry, not with a length depending on the category. And so, okay, so these are the way we are implementing. We have implemented our solution and solution of the state-of-the-art Goldman. So it's a hybrid solution to make JPEG to be able to use JPEG as a real DNA coder, rather than converting, transcoding the JPEG bitstream into a DNA code. And okay, so what happened is that it is really the experiment we did. The results are fresh, so they are. So we need to study more. It's a scoop for you today, so we have to study more and etc. But we put a noise, one noise in the sequence we generate. So it's one deletion. So on the right side we have the JPEG DNA. So with ternary Huffman, Goldman, and pair code inside. So if we lose one coefficient, what we are able to decode. In the middle, it's a transcoding, so it's JPEG providing a binary sequence, and pair code our solution after. And so we are losing one bit and sorry, one nucleotide with a deletion. So we're obtaining this. And on the left is the proposed solution. So the proposed solution that I showed you previously and that we have tested with a web lab experiment. And if we are losing one nucleotide, and this has been the nucleotide has been lost in the LL subband here, the result we can obtain is this one. So there are some black holes. So we can claim here that with this implementation of JPEG DNA, we are more robust. But this is true because we are using a fixed length code. On the contrary, if we are looking at the pure performance in terms of rate distortion, JPEG DNA and transcoding are better than ours. So we have proposed a solution which is not very efficient, maybe in terms of rate distortion. So for a given rate, we don't have the best distortion. Or for a given distortion, we don't generate the best rate, but we are able to be robust to noise. So the idea is maybe to trade off between the solution we propose and such a solution using JPEG or JPEG 2000 that are more efficient in terms of compression or rate distortion optimization, but less efficient, maybe less robust to the sequencing noise. So in conclusion, so we have presented a new encoding algorithm for the robust encoding of digital image into DNA. And contrarily to the state of the art, this solution allows to control the trade-off quality cost, even if we are not providing the best trade-off, but we are able to control this cost, this trade-off, meaning that for a given quality, we are able to know the amount of oligo that we can generate. So this is very important compared to transcoding solutions. Our solution can be applied on any kind of input data, so on binary streams, on symbols, on quantized samples, which is not the case for the state-of-the-art or for all the state-of-the-art solutions. The solution we propose tackles the problems of biological constraints, of course, because we need that for being able to decode something after sequencing. Anyway, it is not efficient in terms of rate distortion compared to a standard codec, and but the standard codec is less robust to noise. Okay, so future work. So of course, it is important to improve the coding decoding performance, and especially if we want to insert the DNA code inside the whole compression system. And what is interesting, I guess, is to explore JPEG solutions. Has JPEG 2000, or maybe one day propose a new solution, new efficient solution. The fact is that if you want to explore JPEG solution, one point very important is to robustify the solution to sequencing noise. And maybe this will reduce the performance of the JPEG codec. Also, if we can generate longer oligos, it will be better, because as we saw, in each oligo, we have to insert a lot of information according to the sequencer, according to the headers, the position of the payload in the data. So this has a cost, and bigger the oligos, the better, because we will have more space for inserting payload. So the amount of the payload will be bigger than the amount of the extra information that I have to insert inside the oligo. One thing very important also, and this is the future of sequencing, I guess, is to use nanopore sequencer. So nanopore is a device like that. This is a very small device that can be plugged on a USB port of a laptop, for example. And you insert your DNA inside this device, and you are able to do the sequencing thanks to your laptop. So this is very interesting. This has a lower sequencing cost, but it is prone to errors. So the errors introduced by this kind of sequencer is bigger than the errors, it's higher than the errors that is introduced by the Illumina sequencer that we use for our wet lab experiment. So we have to deal with new solutions for robust decoding, and maybe why not using a machine learning solution for doing robustifying, or trying to recover the good oligos after sequencing. Okay, to end my presentation, I want just to present the OligoArchive project. So which is an European project, and started last year. So we are working on this topic since 1 year with the OligoArchive. The OligoArchive, the partners are University Côte d'Azur, CNRS, Euricom, so here in Sophia Antipolis, Imperial College in London, and Elixworks, which is a company doing DNA synthesis. So thank you for your attention. I think that I take more time than was allocated to me, so sorry for that. So thank you.
Thank you very much, Mark. It was really fascinating and perfectly targeted to the activities that JPEG is exploring right now. You have been a pioneer in this field, and we really appreciate that you took time to share with us some of the activities you are doing. Now, because of interest of time, I propose again we hold the questions for the end of the session, and we move to the third presentation by Raja to tell us the other side of the story with the micro forms and the macro molecules.
Yes, thank you very much, Suraj. Am I audible, clearly audible?
Yes, we hear you.
Okay, perfect. So tell me if you can see my presentation.
We also see your slides.
Perfect. Okay. So thank you for having me. I'm an assistant professor in Euricom, as Mark already mentioned. And today I'm going to talk about some issues in archiving digital data for a long time, whether it's using analog media or biological storage media. So I'll try to take the standpoint of a digital preservation person here, or a person who wants to archive some data for a very long time period. And I'll actually walk you through a series of problems that such people can face. And very quickly, I will try to introduce you to problems that actually transcend storage, that are beyond DNA storage, that apply to DNA storage also. And I'll talk about some solutions that we are working on. Okay, so just one second. Yes, so this is the typical storage hierarchy that most of us are familiar with. Basically, today we have multiple types of devices that vary in terms of both access latency and storage cost. So here I'm showing you the most popular devices that we have today. The x-axis shows the access latency, and the y-axis shows the storage cost. Typically, if you talk to an enterprise, any company, they organize data in terms of storage tiers. So you have a performance tier where they are storing data using SSDs or DRAM. There is a capacity tier where they are storing data using hard disks. And there is an archival tier, which is what's very relevant to us here, which is intended for storing data for a very long time. And that typically the king in archival is tape. Tape today provides the lowest cost per gigabytes for data archival, and it's at least an order of magnitude cheaper than hard disk drives. And pretty much any large-scale data generation facility, whether it's particle accelerators, whether it's the cloud computing providers, whoever you can think about, pretty much every large-scale data storage facility uses tape in one way or the other. Now, what's the problem with tape? This is a survey I'm showing from the Storage and Networking Industry Association. It's called the 100-Year Archive Survey. Basically, the SNIA went around asking enterprises, museums, a whole bunch of people about their data requirements, particularly about how long do they store data and what fraction of people store data for very long time periods. What's an important takeaway from this survey is that 60% of archival data is stored longer than 20 years. And this fraction of data that needs to be kept long is increasing due to the need to meet various regulatory compliance requirements like Sarbanes-Oxley. For various legal issues, people need to keep data for longer and longer time periods. And Fernando talked a little bit about memory institutions, institutions that have to preserve culture, institutions that have to preserve journals and all kinds of important documents over essentially forever. And these institutions, some of these were also a part of the survey. The problem here with tape is that we have a limited lifetime of 10 to 20 years. But it's not that we have not had media which can last longer than that, right? So I'm giving you an example of two media types here. One is film, microfilm. Microform has been used by libraries and museums for ages for preserving journals, e-journals, for actually taking journals from hundreds of years old, mapping them into a very compact form factor and storing them for hundreds of years for the future, right? And today, if you look at preservation standard LE 500 microfilm, it's rated to last for 500 years. And even going back even older than that, we have had, for several millennia, we have had paper, right? And we have been preserving knowledge on paper. And today, ISO 9706 permanent paper can last for 500 to 1,000 years. So today, many libraries, many museums, many memory institutions actually use this media in one way or the other, both microfilm and paper. It's just that we don't hear about it because it's not very popular. So an interesting question, of course, is can we use this media as a digital data storage media? Of course, the answer is yes, right? You can, and the way to do that is to convert, you can take your digital data, which is a sequence of bits, convert it into a sequence of barcodes, and you can then print these barcodes to paper or shoot these barcodes to film. And if this sounds like something that's fairly straightforward to you, the fact is that there are companies today, and I'm sharing an example of this. There's a company called Upalia, which is a startup here in France, and Eurecom, I'm collaborating with them from Eurecom. They have a system called MicroOlonus, which can actually save digital data on paper. And they're talking to many institutions here, including the nuclear waste management facilities and a few others. The point is that they can actually store about 50 kilobytes of data per page of an A4 paper, or they can store 1.3 gigabytes of digital data in a 66 meter film. So we have had media which can last long. The problem with this media is that physical density is severely limited, right? So we don't have a media that can last for 500 years and is also extremely dense, as dense as tape, or maybe even denser than tape, that we can pack a lot of data on this media. And that's exactly where DNA comes in. So I'm not going to walk into too much details because Fernando and Mark did a great job explaining the role of DNA. The top graph actually is taken from the Semiconductor and Synthetic Biology Consortium, and they basically shows you the potential that DNA has in terms of density, because it has 7 orders of magnitude higher density than tape, which makes DNA a fantastic, highly dense 3-dimensional storage medium. And of course, the bottom figure shows you an experiment from Professor George Church's lab on trying to resurrect the woolly mammoth, which is an extinct species that lived about 5,000 years ago, from the fossils, right? So if DNA can last in such adversarial climatic conditions, DNA stored inside an imagined DNA shell capsule can last for thousands of years without any problem. There is no problem with that. So we do have now a media like DNA, which is dense and can last for a very long time. And this is exactly the focus of Project OligoArchive, as Mark mentioned, which is how to store digital data on DNA. And we are looking at pretty much the entire stack, right? So we are thinking of DNA as a disk, or thinking of DNA as a tape, and looking at the entire stack, starting from the media layer where Mark talked about synthesis and sequencing. And I'm also working on various encoding techniques for storing other types of data, databases particularly. And we're also looking at the controller layer, where we are looking at once you have data stored in DNA, can you directly execute SQL queries, for example, on DNA using enzymatic reactions? Our partners in Imperial are looking at this. We are looking at the operating system layer. Think of a OS over DNA, which gives you the ability to read and write individual files. So we are using primers, sequencing primers for this particular purpose. I won't go into details about this today. But essentially, the project focuses on all these aspects. But the density problem and the longevity problem is only one of the issues facing long-term data storage. The second problem facing long-term data storage is what people refer to as, just give me a second, I'm just going to move this away. Yeah, what people refer to actually as the media obsolescence problem. So the figure on the left-hand side shows you the rate of improvement of areal density of 3 storage media. The red line shows you the improvement in density of disks. The green line shows you the improvement of NAND flash, and the blue line shows you the improvement in tape. Now, as Mark mentioned, due to various limitations in magnetic recording technology, disks have actually hit a blockade. They have hit a wall, and they are not able to improve density at 40%. So the Kreider's rate has stopped. The rate of improvement following Kreider's rate of disk drives are stopped. NAND flash is using volumetric scaling densities, 3D stacking and techniques like that. So because of this, NAND flash is improving at a much better rate than disks. But of course, flash is way more expensive than tape for storing archival data. So it's not relevant to us here. What's interesting here is that tape has an average improvement rate of about 30% per year. So the tape density is improving quite well. And for the foreseeable future, tape is going to improve in density, meaning the tape is going to become cheaper and cheaper as we go forward, at least for the next foreseeable future. The problem with this is the fact that if tape density improves at just 10%, let's say, right, not even this 30%, just 10% per year, in 10 years, you will be able to store the same amount of data that you're storing today in much lesser capacity, right? And this basically means that people will be migrating from one generation to another generation just to save space, because storage space in data centers is a premium commodity. So people are going to migrate. And this is actually a negative side of Kreider's rate, right? It's the fact that because of the fact that storage media improves in density, there is an incentive for people to actually migrate from one generation to another. Associated with this is also the fact that the LTO, the Linear Tape Open Standards, Linear Tape Organization, it actually, the set of backwards compatibility provided by tape drives is limited only to 2 generations. So the figure here shows you this diagonal pattern of compatibility, as you can see. So if you have a LTO4 reader, there is a backwards limit of up to LTO2 that you can actually read. So you cannot read LTO1 tape. So this basically mounts on you even more pressure to actually migrate data, right? And clearly you can see tape suffers from media obsolescence, meaning after a few years, every few years, the data that you have on tape today is going to be obsolete, because the tape itself is going to become obsolete, and you have to migrate from one generation to another generation of tape. you might actually think that DNA solves this problem, but the problem is slightly more insidious. The issue is that DNA and paper solve the tape reader problem, right? So remember, we discussed that tape readers support backwards compatibility only 2 generations, right? But with paper, all you need to read paper is a basic scanning technology. So you can imagine that in the foreseeable future, any scanning technology will be able to read data from paper. So you don't have the reader problem. Similarly, if we store data on DNA, we will always have the need to sequence human DNA. This is just going to be the case because DNA is being used more and more for precision medicine and various other applications. So we will always have the ability to read DNA. So if data is stored in synthetic DNA, we will always be able to read it back. The problem is not with the reader. The problem has to do with decoding, right? Getting data from paper requires converting the barcodes back into digital data. And getting data from DNA requires converting the reads, the sequencing reads that Mark and Fernando mentioned. You need to convert them back into digital data. The decoders that I use for this purpose, as you just saw in the presentations, these decoders are extremely complex, right? So they can use highly optimized compression algorithms. So you need to have the decompression versions of these, and they must use error correcting codes. So if you're using Reed-Solomon codes, or if you are using low density parity check codes, you need to keep around a parity check matrix, and you need to keep around the parameters that are associated with the decoder for decoding. So the question then here with obsolescence is, how are you actually going to preserve these decoders? If you store data on DNA and 1,000 years later somebody comes, gets the reads out of the DNA, how are they going to be able to decode it back into the original digital data if they don't have access to the decoders? Without the decoders being preserved, your media is essentially obsolete, right? You cannot make sense of the data. So the solution to this problem that we have been working on is one that comes from a field called digital preservation, where a whole group of people over the last few decades have been working on this problem. And the central idea is to use emulation to archive decoding logic. Now, for those of you who are not aware, emulation is a technology that's used to simulate one hardware environment using another. And typically emulation is very popular for getting old games like Atari games to actually run on modern hardware. These days, emulation is also being used by various museums and libraries to get very old software artifacts, like certain interactive books that were developed 20 years ago or 10 years ago that do not work anymore because they were developed for a particular platform, for getting these to actually work because they're cultural artifacts, getting these to run on today's environment. So the idea is to actually use this emulation technology to preserve decoding logic. And the way you do this is quite interesting. What you want to do is you want to use an emulator to actually build a virtual software processor which has its own instruction set architecture. So this processor is not real at all. It does not have to resemble anything like the x86 processor or the ARM processor. It can be a very simple reduced instruction set processor with just 10 or 20 instructions. And what you will do is write the decoders, or you could basically, if you have the tool chain, compile the decoders written in C into this ISA, right? So ultimately you get your decoders into a set of assembly instructions, the assembly instructions targeting this virtual processor. And then now you have your decoder as data. So you can store the instructions of the decoder together with the data. If you have to restore the data, what you have to do is you need to make sure that this emulator is around, right? So the decoders actually don't need to be kept around. The emulators need to be kept around. And if the emulator is simple enough that it supports only a few instructions, you could potentially have someone implement this emulator 1,000 years later using a pseudocode described in a page of text. So you can have one page of text describing some pseudocode, and any person in the future can take this page of text, can take the pseudocode, implement it on any language, any runtime, any OS that does not even exist today, right? And then create this virtual software processor, which will then automatically load this decoding logic, which is written using the software processor, execute the decoder to convert the data back. And this is exactly what we did in a very recent collaboration with Upalia. And we demonstrated this using databases. So I'm going to give you a very quick overview of how this works, now not going into too many details. So this year, let's say in year 2020, we take the database and we dump it out into a file. And then we pass it through Microlunus, the software that we have developed, we are collaborating on. And that outputs a series of barcodes, okay, which are basically images that you get, and a text file, basically a page or 4 pages to be precise, which contains the pseudocode, the algorithms for implementing, for bootstrapping a software processor. And this is, you take this and you can print it out into microfilm, you can print it out to film or paper. After, let's say 100 years or 1,000 years, a user is going to scan this using any basic scanning technology, and they get the barcodes back and the pseudocode back. Using the pseudocode, they are going to implement this bootstrap, which will actually create a virtual software processor. The processor will automatically load the instructions from the barcodes corresponding to the decoder, and the decoder will then convert the data back, and you can load it into a database in the future, 100 years into the future, without having to depend on any technology that exists today. Okay, so we call this method universal layout emulation, and we demonstrated this end to end. And we just wrote about this, a paper about this. I'm putting a link to the paper here. And we are now looking at extending universal layout emulation to DNA data storage, right? So this solves the problem using ULE and using our system. You solve the problem that you can preserve the decoders without having to rely on any existing technology, without even having to assume that the emulator needs to be preserved, right? So that's fantastic. But unfortunately, that's not the end of the story. Suppose you actually recover an image as a binary file in 1,000 years, right? The question then is, suppose you actually convert it back. You have the decoder, you convert it back, and you get the JPEG file out. Now the question is, if you have a JPEG file, how can you be sure that anybody can see the file? How can it be rendered, right? So the problem here is that you have to now not only preserve the decoder, but in order to make sure that the format, the file format that you're talking about, JPEG, is actually available in the future, you need to actually preserve the rendering stack. You need to preserve the tooling, tool chains that are used for converting JPEG format into some other format. A whole lot of it, the entire software environment needs to be preserved. One way to do this, which is definitely being used by many people working in digital preservation today, is to actually build on emulation, right? So instead of emulating a simple software processor, build an emulator for a very complicated CISC processor like x86. Take existing programs today that can work with JPEG, put them in the emulator, preserve the emulator, make sure that people are actually porting this emulator across generations of processors. And this way, by simply porting the emulator, you will be sure that even 1,000 years into the future, somebody will be able to load Windows 95 back on the emulator and get their a rendering software that that they used, right? For example. And Olonus, of course, the prototype that I described, micro-Olonus, depends on Olonus, which is actually an emulator. It could potentially solve this problem, the problem of porting this emulator using the bootstrap. So this is one solution. Of course, it's a complex solution. It's complex also because now you have to tell everybody who wants to preserve images, cultural institutions, libraries, that you need the technical ability to run an emulator. Okay. People in digital preservation are working around this by actually using emulation as a service, running it as a cloud service, for example. That's one way, but it's still by no problem. The other solution, which is very relevant here, is to use standardized formats like JPEG 2000. So the assumption here is that if the format is actually standard, and if information about the format is widely available, then it will always be possible to actually build a renderer in the future, even if it's necessary, right? Now, the issue with this is that the ability to read a file in the future is a function of how widely the format is going to be used. A couple of years ago, back in 2013, I'm quoting here a blog from Chris Adam, who was the tech lead of World Digital Library at the Library of Congress, the project World Digital Library. And there, the question they actually looked at when JPEG 2000 was around at that time, the question it just was new in the blog and everybody was looking at it. The question they asked was, is JPEG 2000 a preservation risk? So some of the points that they actually mentioned in that particular blog, I would like to bring it out here because I think it's a very interesting question. They are very interesting questions that we should also be asking, given that we are looking at a standard for JPEG for DNA, right? So for example, some of the things they mention are that the first thing, of course, they mentioned is that JPEG 2000 depended a lot on proprietary software at that time. Browser support was absolutely required, important for them because a lot of applications needed to access the image from the browsers, and only a few browsers supported JPEG 2000. Complexity was a major issue for libraries, and here they gave an example where the OpenJPEG, they compared the lines of code of OpenJPEG and the entire Python, sorry, it's not Imagine library, it's Python image library. there was a huge disparity, and OpenJPEG was incredibly complicated in terms of the code base. And this was an issue for them in terms of preservation, because they were looking at preserving important historic images, documents in terms of images, using JPEG 2000, for example, right? Now, if you fast forward 7 years, today JPEG 2000 is definitely used by many memory institutions. But if you look at the state of the art, there is still the problem, and it's also in this blog. A few people commented just recently that on Linux, very few open source programs properly process JP2 files. And in fact, a very recent article, which is entitled JPEG 2000, a guide for digital libraries, openly says that if you're serious about deploying JPEG 2000, you need to get a good quality commercial codec. Do not rely on open source implementations, right? Now, the takeaway that I would like to emphasize here is, I think JPEG 2000, of course, is actually being used widely. It's a fantastic standard, and more and more initiative is being put by cultural institutions and memory institutions in adopting it. But I think it offers a very good lesson in terms of format preservation. So if we are going to use the second solution, which is relying on formats for solving the format obsolescence problem, then we have to make sure that the standard for long-term storage must have enough open source tooling, and it will have enough open source tooling only if it is extremely popular, right? And it will be popular only if it is adopted by many people. So these kind of things are linked to each other, and I think it's important. I'm just bringing the perspective from the community of people who work on archival or digital preservation. So to sum up, I discussed three main issues in long-term preservation. I talked about media decay, which is quite often what we actually talk about when in terms of DNA storage, right? We always talk about the fact that tape does not have a long lifetime, but we have to remember that there are also other media which can last long. So the second problem is media obsolescence, right? The fact that you cannot get data back, recover your data back if you don't preserve the decoders. You could solve it with a technique like universal layout emulation. And of course, the third problem is format obsolescence, for which pioneering work done by JPEG needs to be done here also. But we need to make sure that the standards are simple enough, the standards are popular enough that they lead to a wide selection of open source tools. Having said that, there is a big ticket issue that I have not covered here, which is money. It's well known in the digital preservation that storage costs can get out of hand and will get out of hand. Tape is already extremely expensive due to the continuous migration that I talked about. DNA synthesis today is orders of magnitude more expensive than tape. There is definitely promising enzymatic synthesis technology in the horizon. But something to keep in mind is a traditional rule of thumb that has been adopted by digital libraries. I'm borrowing this from blogs written by Dr. David Rosenthal, who's an expert in digital preservation, and he's the creator of the Lots of Copy Keep Stuff Safe program in the Stanford Digital Libraries. So generally in digital preservation, it's well known that half of the cost is for ingesting the data, which is getting it into the preservation system, and the other half is actually preserving it. And in the preservation part, 1/3 of the cost goes to storage and 1/6 of the cost goes to accessing it back again. Because once you preserve the data, if you need to provide access to someone, you need the technology for that as well, right? The question here is this the case for other parties today? Is this the case for cloud storage, cold cloud storage like Facebook, as Fernando mentioned? Is this the case for enterprises, state and private archives? Or is it something that's only specific to digital libraries? The reason why this is pertinent and relevant here is because there is a cost complexity trade-off that we should think about. If we store 50% less data using these cost metrics, we still only save 1/6 of the overall cost, but with a much more complicated compression scheme, right? So the point here is that if this is actually, we need to know exactly the cost breakdown of how much does it cost to preserve data in a particular application domain, and based on that we can actually decide whether what type of technology to actually adopt for standardization or for preservation, for example, right? So having said that, I thank you all for the opportunity that given here, and I'll take any questions.
Thank you very much, Raja. So let me, at this moment now, stop the recording. This way, probably people will have a little bit of a better feeling to ask questions. So